When we're talking about temperature, and we know now that most species perform best in a fairly narrow range of temperature, then when we look at the individual organisms and we can look at how the environment affects them, we can see that they are changing their behavior. They can be changing their ability to grow fast. They could affect how many offspring they produce, escape predators, catch prey. So all of these things put together is looking at the overall performance of an organism. Now, when we look at the performance at a molecular level, a biomolecular level, you'll remember from your general biology class that temperature is going to be affecting enzymes. Enzymes are proteins that help speed up chemical reactions. So enzymes usually work best in a limited range of temperatures. Most enzymes at low temperatures have a very predictable shape. The problem at a low temperature, your enzymes are shaped perfectly to help speed up your chemical reactions, but everything at low temperatures is moving kind of slowly. So remember, the molecules at low temperatures are moving more slowly, and so that will cause your chemical reactions at the temperature is low to be a little bit slow. So enzymes usually work better as they are warming up. But when you get to an excessively high temperature, remember your enzymes, your proteins will denature so that they will start to lose their proper shape. They will not be able to bind with the substrates, and they won't have the flexibility to do their particular functions. So there's an optimal range of temperatures for most enzymes. So if we look at, and we're going to be looking at this with a number of different examples, but if we look at how enzyme activity varies with temperature, remember on a graph, your x-axis, your horizontal axis, is going to be your independent variable. So the enzymes are not making the temperatures hotter or colder. The temperature is actually influencing the enzyme. So the enzyme activity is dependent on the temperature. So on the x-axis, I'm going to put temperature. And in this case, I'm not going to put specific numbers on here because that's going to vary with the organism. We're just going to basically indicate that you're going from a low to a high temperature, so you know what direction this is going. And then on the y-axis, I'm going to put some sort of a measure of my enzyme activity. So the enzyme activity can be measured in a number of different ways. For instance, if we were looking at photosynthesis, we might be measuring the enzyme activity based on the rate that carbon dioxide is taken into the plant for photosynthesis. When we look at the relationship then between temperature and enzyme activity, the ability of the enzyme to work, we know that if the temperatures are low, the enzyme really, everything's kind of moving sluggishly. When the temperatures start to increase, we can see that the enzyme activity is going to get higher and higher and higher. But then we start to reach a point where the enzyme will start to denature. And when we reach this point, we can see that the reaction rates will start to slow down. You'll reach a peak, and then it'll denature and fall apart and just not work anymore. So you get a curve, and we see these over and over again. And if you flip through your chapter, you will see these curves that show the enzymes will be working together or working better as temperatures increase. So this is increased movement rates of molecules with temperature, and this is the denaturing. So your enzyme starts to fall apart. These enzymes, remember, they're proteins. Proteins are made using the instructions on the DNA that your parents gave you. And so they must be important if that's what your parents gave you. That's all they gave you were the instructions to make proteins. And so these enzymes have to run all your chemical reactions in your body. Your body is such a low temperature for this amazing chemistry to take place that without these enzymes speeding things up at these low temperatures, you would not be able to complete any of your chemical reactions in time. You would not exist. So we need these enzymes to make everything else work within our bodies. So they then give you a really nice example in your textbook of looking at how these enzymes perform at different temperatures. And this was the work by Baldwin and Hochachka, looking at the enzyme acetylcholinesterase in rainbow trout. Now, let's say you have forgotten anything about acetylcholinesterase and acetylcholine, and you don't remember anything about enzymes. This is a good time to go back, look these things up. So we've assumed that you have some general biology and biochemistry knowledge about these things, and if you don't forget, go look these up. Make sure that you understand them. So acetylcholinesterase is an enzyme that's produced in the synapse between your neurons. So remember, in your nervous system, you will have the little spaces. So you have a chemical signal going down your nerves. Let me get a brighter color. You have chemical signals going down your nerves. And then when we're going to go to the next nerve, we have a little break between nerve cells called a synapse. And so we got an electrical signal, so we're going down the nerve this way. It hits the synapse and it has to jump, and the electrical signal can't jump. So what you do is you produce a chemical signal, and so you're going to produce acetylcholine from this one nerve cell, will travel over to this other nerve cell, which will trigger it to continue the electrical signal down that nerve cell. Now, we can't have this acetylcholine just continue to sit here and making things just keep going when you don't need them going. So you need an enzyme that's going to come in and break down that acetylcholine. And when it breaks down that acetylcholine, then you can stop, you can relax. Okay, I don't have to keep this nerve tickling all the time. And so acetylcholinesterase is what is breaking down that acetylcholine in the synapse. So what did they find? So once again, when you get to something like this and there's a figure with it, and in this case it's going to be figure 5.8 in your textbook, and they describe your experiment and how they did it, read over these experiments, and I expect you to be able to do that. So Baldwin and Hoekstra in 1970, they published a paper about studying the influence of temperature on the activity of acetylcholinesterase, an enzyme produced at the synapse between neurons. This enzyme promotes the breakdown of the neurotransmitter acetylcholine to acetic acid and choline, so turns off the neurons, a process critical for proper neural function. The researchers found that the rainbow trout produced two forms of acetylcholinesterase. One form has the highest affinity for acetylcholine at 2 degrees, which is the winter temperature of the water, 2 degrees Celsius. However, the affinity for this enzyme for acetylcholine declines rapidly as the water warms up over the summertime. And there's a second form. They produce two different forms of the same enzyme. They're so clever, these fish. The second form of acetylcholinesterase shows highest affinity for acetylcholine at 17 degrees Celsius, which is in general the summer water temperature. The affinity of the second form of acetylcholinesterase doesn't work well at a lower temperature or a higher temperature. So they have two forms of the enzyme, and it works at two different temperatures. So how is this going to look? We have temperature as the independent variable. And we have how well the acetylcholine works. So your enzyme substrate activity, so how well the enzyme will actually bond with the acetylcholine. So, and we're going from low to high temperatures here, and low to high activity there. It's amazing how many times on an exam people will draw a graph and they won't label low and high, and they'll do it backwards and the graph looks backwards. Sometimes they will put the dependent variable on the x-axis instead of the independent variable. So this is the independent variable. This is the dependent. It's very hard to determine, interpret it if it's mixed up. So, and actually in this case, I can put some numbers on here. I can put 0 degrees, water's frozen, obviously nothing's going to be working then. And then I could put things at 10 degrees Celsius, so this will be Celsius, 20 degrees Celsius, and 30 degrees Celsius. So I know there's 2 forms of this enzyme. So I'm going to draw 2 lines on here, one for the first form, one for the second form. The first form, as it said in the description, has a very high affinity, works best at 2 degrees Celsius. So at 2 degrees Celsius is where it's going to peak. So I can take this first form, and I'll call it the cold water form, is that when it just gets above freezing, it's going to start working better and better as it gets warmer and warmer. And then, actually, I'll make this one, this is the cold one, so I'll make this one blue. And then when we get to 2 degrees Celsius, this is where it's at its peak, and then it'll start to degrade as it gets warmer, and then it drops off as it gets warmer and warmer, as the protein denatures. As that one enzyme is starting to fail, then the second version of this enzyme is going to start to kick in and start to improve, going faster and faster. And that one peaks at about 17 degrees Celsius, and then it will denature. So we have 2 different forms. We have the form that is peaking at 2 degrees Celsius, and we have the second form that is peaking at 17 degrees Celsius. So we have two curves for this. And that's the picture from your book, which is much better than the one that I drew. But you should be able to reproduce something like this on an exam question. So we want to move on to talk about plants, but they do give you another example of animals in your textbook with a different way of measuring metabolic activity, and that was the eastern fence lizard, which we do have in New Jersey. So make sure you read that over, and they are looking at the performance of the animal in terms of its rate of metabolism of food. I'm going to move on now to talk about how plants are also influenced by temperature through the process of photosynthesis. So this is obviously a chance for you to review what photosynthesis is. And this example is looking at 2 plants that have evolved to grow in very different environments. We have a moss growing in the boreal forest that is growing on the forest floor, so it's a very cool area that it is growing up north and in the shade, versus a desert shrub, which is Atriplex lentiformis, and they talk about this desert shrub various times in the textbook, so it might be important to kind of get interested in this or familiar with this shrub. And once again, with these plants, we can see they're adapted to very different environments. So on the x-axis, we have the temperature, which is in degrees Celsius. That's the independent variable, going from very low temperatures to high temperatures. And then on the y-axis, we have the photosynthetic rate. And instead of giving you the actual rate, maybe of CO2 intake or something like that, it's doing it as a percentage of their maximum, because obviously this desert shrub that is out here in the sunlight in the middle of the desert has a much higher photosynthetic rate than the moss, which is sitting in the shade in the forest. So they want you to be able to compare these the shapes of these two graphs and where their peaks are. And we can see the boreal moss shows exactly the same patterns we saw when we were talking about the enzymes in the fish, that as the temperature is increasing, the photosynthetic rate is going up. Its peaks as it starts to denature, so you find the peak at 15 degrees Celsius, and then it'll slowly start to break the enzyme down till you couldn't grow that moss at a very high temperature. And even if you took that moss and decided you were going to take good care of it and grow it in your house is at 22 degrees Celsius, and it's not going to do as well in your house as it was doing out in the forest floor. This is in contrast to this desert shrub. The desert shrub really could not grow at 15 degrees Celsius. Its enzymes are not really going to kick in until it gets much warmer than that. And then they peak at 44 degrees Celsius, which is incredibly warm. And then they will rapidly denature after that. So they're not going to be able to function at a much higher temperature. So obviously these are genetically adapted to thrive in the environment. They're very different environments, the boreal forest and the desert biomes. If you're asked to draw the photosynthetic rate of a moss from the boreal forest and a shrub from the desert in relation to temperature, you know to put the temperature across the x-axis. That is the independent variable. And in this case, you could probably even put some numbers on it. You could probably put 0 degrees Celsius and 50 degrees Celsius, or 60 degrees Celsius across there. Or at the very least, you could put low to high. And then on the x-axis or the y-axis, we have the dependent variable, which in this case was the percent of photosynthetic activity. So percentage goes from 0 to 100. You have one line for the moss. You have one line for the shrub. Actually, let me erase those. One line for the moss. They have to go to 100%, right? And that 100% for the moss was around 15 degrees Celsius. And then the shrub, we know, started around there. And the shrub peaked at 44 degrees Celsius. So you even have a couple little numbers you could put on this. And there you go. Now, when we've been talking about adaptation to different environmental conditions and temperatures, we've been talking about genetic adaptation of populations. However, we can also talk about adaptation of the individual, and that is acclimation, where you have a short-term physiological adjustment, and that allows you to respond to the temperature. So this study is the same plant we were just talking about, that desert shrub, the Atroplex lentiformis. And this was done to see if it could acclimate, if it grew in different temperatures, if it could change its photosynthesis. And this was a work done by Robert Percy. And he clearly demonstrated the effect of acclimation on photosynthesis. So he took a population of the desert shrub that was growing in the Death Valley in California, a very hot environment, and he took cuttings of them. So he had cloned plants, propagated plants from the cuttings so that they would be genetically identical clones. And the clones from the Death Valley plants then were grown under two different temperatures, one in a hot set of conditions at 43 degrees Celsius during the daytime and 30 degrees at night, and the other was under cool conditions at 23 degrees during the day and 18 at night. And then he measured the photosynthetic rate of the two plants. And as you can see, the plants that were grown at the daytime temperatures at 43 degrees had a much higher peak in their rate of photosynthesis. So they were peaking at around their daytime temperature over 40 degrees Celsius, and the clones of the shrubs that he grew at the cooler temperatures were peaking. It's still higher than the temperature that they were growing at, but it was much higher, much lower. So it's about a 10 degree Celsius difference in the maximum rate of photosynthesis between the two different populations. So this is a physiological adjustment, and so we know that individuals can acclimate to different temperatures and adjust their physiological responses. So the enzymes that were working at one temperature are different than the enzymes that are working at different temperatures. So this means that plants and animals can acclimate to the changes in temperatures with the seasons. We saw this with the enzymes of the acetylcholinesterase and the salmon, and now we see it with the photosynthesis and these. And we know in this case, how do we know that these were not genetic differences? Remember, he took clones. He took genetically identical individuals to do this study. Now, we have been talking about photosynthesis. I wanted to take a moment to review what photosynthesis was in case you've forgotten. So remember that the plants have to take in carbon dioxide because they're going to, in photosynthesis, they're making sugar. So we need carbon for sugar, so they're taking carbon dioxide. Where's that carbon dioxide coming from? Well, that's sitting out in the air. How does it get into the plants? Into the leaves. The plants don't have lungs, so into the leaves of the plants. So the leaves of the plants have little holes on most of them on the underside, called stomata or stomata, and that allows the carbon dioxide to come in. They will also need water. The water is in the soil. So that's sitting down there in the soil, so it's going to come into the roots of the plant. So we're going to have the roots down here in the soil. It's going to come into the roots, and then it's going to have to go up the stem. And when it gets to the top, it's going to go onto the leaves where it can mix with the carbon dioxide. And we can go through a whole series of chemical reactions in the presence of sunlight. So remember, we are also going to need the sunshine here. So we have the sunlight, which is a source of energy. And through the process of the light-dependent reactions, that sun is going to split this water. So it's going to break this water up, and when it does that, it's going to take the electron from the hydrogen, and it's going to heat that up, and we're going to use that electron to generate some ATP, and the plant is going to get rid of the oxygen. So the oxygen is a byproduct that is released, and that's released back out into the air through those very same stomata in the leaf. So it gets rid of the oxygen. We still have the hydrogen. We're going to make sugar in the light independent reactions. So remember, the sugar is the glucose, it's C6H12O6. So we're going to produce some glucose there. And that glucose can go and be, so we use the ATP, remember, from the sun and the electron and the hydrogen, so that glucose has been glued together with all of that energy. That energy can be used in cellular respiration then, so we can use that energy to for the plant to do its chemical reactions, to grow, and we can also store it, so we can put that glucose. So remember, this is glucose, so we can use it, or we could store it, and the plant will make long chains of glucose, and which is starch, which they could take down and put into their roots and use later. So it can store as starch, which is chains of glucose. In order to balance this equation, though, you're going to have to take in 6 carbon dioxides, and we're going to have to take in 12 waters in order to make all of this work. And you're going to get 1 molecule of glucose out of that, but you're going to have 6 molecules of oxygen gas that you release. And then you're also going to have to take, remember we were ripping apart this water and we've got some hydrogen left over from that. And so we're going to take that hydrogen and we're going to put that in the form of water. So that's an H2O. And so that water is going, there's going to be 6 of those produced as well. 6 waters. And that water is also going to go out into the air through the stomata and the leaves. This release of water into the air is significant. This is the evaporation of water into the air, so this is going to be water vapor. And this process of evaporating water from the leaves of the plant is called transpiration. And if we're talking about the evaporation of water, whether that's sweat or whether that's from the leaves of the plant, that's going to cool the plant. That's going to affect your temperature as well. So that is going to have a kind of a cooling effect. Okay, so we do know that extreme temperatures are going to affect the enzymes in photosynthesis. So here's the equation I just wrote for you. Extreme temperatures will usually reduce the rate of photosynthesis, and this is an issue for global climate change because we're finding the tropical rainforests are getting so hot, the trees are not able to complete photosynthesis. The enzymes are breaking down. And we know that the plants have different optimal temperatures. So we've talked about animals, we've talked about plants. Let's go back to looking at microbes. And there are microbes that seem to live in all temperatures around the world. You can find them in cold waters of Antarctica, the boiling waters in the Yellowstone hot springs. However, these extreme environments have specific microbes that live there. There's no microbe that can live in all the environments. Marita studied the effect of temperature on the population growth on the bacteria that loves living in cold water, cold-loving or psychrophilic marine bacteria around Antarctica. And he took these bacteria and grew them in a temperature gradient incubator for 80 hours. And during that time, he ranged the temperature from -2 Celsius to over 9 degrees Celsius. And he found that these bacteria grew their fastest at 4 degrees Celsius. So even though if you got the water warmer, it wasn't going to make them any happier. He even found that they were able to grow in super cold water. Remember, salt water is going to have a lower freezing point. He could even find that he could grow it in super cold water at -2 degrees Celsius and even some of the bacteria were growing at minus 5.5 degrees Celsius. On the other hand, some microbes can live at very high temperatures, and they have found microbes living in all of the hot springs that have ever been studied. So these ones that love the warm are called thermophilic, and they can grow at temperatures above 40 degrees Celsius is how they're defined. And so many of the heat-loving microbes can only live in those temperatures, so that they are hyperthermophiles, which means they have temperature optima over 80 degrees Celsius. And some of them can even grow as high as 110 degrees Celsius. So the most intensive studies of these have been done at Yellowstone National Park, and they were able to see that even some of these guys were able to get their energy by oxidizing sulfur as opposed to using the energy from the sun. And it's interesting because since we know that they could do their chemical reactions at such a high temperature, it meant that they had enzymes that were not degrading at these extremely high temperatures. So we actually use those enzymes when we're doing PCR, when we're doing DNA replication, in order to make sure that we can separate the strands of the DNA. We heat them up, and we don't want the enzymes to denature, but we took the enzymes from these bacteria and have been able to use that. And that's why PCR was possible. And we couldn't do PCR until we discovered some organisms that could work at these extremely high temperatures. So you can heat your DNA up to 90 degrees Celsius, separate the DNA strands, and the enzymes will still stay intact. And then you can cool it and do the replication on each side of the PCR, or each side of the DNA strands. So this is looking at the graphs of the growth of these bacteria. And so we have the bacteria that were growing in Antarctica, and we're looking at on the x-axis, once again, we have the independent variable being temperature, and on the y-axis we have the rate of population growth being our measure. And we can see we have the peak of the population growth in these Antarctic bacteria at 4 degrees Celsius. The second graph here is looking at these bacteria from the hot springs in Yellowstone. Excuse me. And so we can see once again, we have temperature across the x-axis, which is it's our independent variable. And here the rate of sulfur oxidation. So this is the rate that they are using the sulfur for their energy source is being measured. And we find that their peak is around 60 to 63 degrees Celsius. And the hot spring itself was at around 59 degrees. So they're really operating right at their peak amount of peak photosynthetic rate at the hot spring temperature. So that's the end of section 5.3 in your textbook, and you should be able to answer the questions at the end of the concept review questions there. The first one was asking about signs of thermal stress in fish swimming on their sides and swimming in spirals. Explain, using what you know about temperature and acetylcholinesterase, explain what's going on. So think about what acetylcholine does, what acetylcholinesterase does, what happens when that denatures and stops working. So you should be able to answer that question. And then the second one is how can we be sure that two distinctive responses to temperature shown by Atriplex lentiformis were due to acclimation as a result of the genetic differences. And it's